Good evening, Kritika. Oh, we are live. Excellent. Thank you, Karthik. So, thank you, everybody. Thanks for turning up. And before we begin, we were looking at the uh, comments and that you have sent in the feedback, and many of you have requested for transcripts of the lectures. So I think there was an announcement made in the Google group that we have a website running with this course which contains the transcripts of the lectures. It We are also going to put up the audio files of the lectures so that you can listen to them as podcasts. So Anish, who as you know is staying for this course, he is going to put up the links in the chat box. So please take that link and use that. Okay. Some of you have also commented very justifiably that it will be nice to have the links to the various videos that I mentioned on my PPT. So those links are also given on that same website. And in a few cases, I have given some extra resources. So actually right now I'm extremely stuck up with recording these videos, but over time I'm going to put up a lot of extra resources for each lecture, so that at one place you will not only get what I'm teaching, but you will also get to know about other things which you can explore on your own. So essentially, Anish, can you please put up the link to that over here? Just go there and most of the extra information that you are asking for, it will be there. Okay. So maybe what I will do is I will call up people one by one, and then you can ask whatever questions you have. So there is somebody who is simply showing up as user. So I don't know user, I don't know who you are, but could you please unmute yourself, introduce yourself, and tell me if you have any queries? Good evening. So do you have any questions? Not yet? Okay, great. So then we'll move to Aradhya. Aradhya, do you have any
Yes,
queries?
sir.
Yes,
So I
please. Hmm.
just started my curriculum, just shifted to my hostel right now. So I was busy with all the shifting work. So
Okay.
I don't have much doubt yet. I'm halfway through week 2 right now.
Okay, fine.
Next
ठीक है.
step, surely.
Perfect,
Thank you,
perfect.
sir.
Thank you, Aradhya. Prem Kumar, are you there?
Yes, sir, I'm here.
Huh. So do you have
Yeah.
any queries, questions?
No, actually, I have just completed the week three contents. Till then, I don't have any queries. I'll finish and I'll try to put the question in the next interaction.
Okay, fine. ठीक है. Thank you.
Thank you.
Akib, are you there? Akib? Huh. So Akib, do you have any queries that you would like to put forward? Yeah. Darshana, I'll come to you in a moment. Okay. I'm just going serially so that everybody gets a chance to ask their things. Kritika, do you have any question? Kritika? Anish has given the link to the site on the chat in the chat box. So I request all of you to please copy that. Okay. Kritika, if you don't have anything, Khushi, do you have any query?
Good evening, sir. Yes,
Good
sir.
evening,
I do
Khushi.
have a
Huh.
query.
Tell me, please.
So, my question is, suppose if we found out about two species from a fossil,
Hmm.
suppose we call them species A and species B.
Hmm.
So, how we will interpret from the fossils that species A is the ancestor of species B, or species A and B share a common ancestor? Like when we draw a phylogenetic tree, sometimes we branch out two branches and just put both organisms there. And sometimes we just put an organism on one branch, and then to the next branch we further divide and then put one organism. So, like, what, how we
Yeah.
will interpret
See,
that?
if you have just two things, then it becomes very difficult to answer that question. Okay. One way, if you have only two and nothing else, one way is to date the fossils and try to see which one is a more ancestral form. Okay. If both of them are roughly coming out at the same time, then you can say that, depending on the characters, that there might be a common ancestor between the two. But otherwise, the question that you are asking makes more sense if you have like 10, 12 or whatever, more than two, much more than two number of species, and within that you are trying to come up with that answer, right? And there essentially, it entirely boils down to the way which tree making method you are using and so on. And for example, the one that we ended up discussing in the, I think week 2 is when we discussed it, right, was the parsimony method. So the parsimony method may then you again use the parsimony thing and see which one is coming up as an ancestral form and which one is coming up as a whatever, branched later kind of a form. So that is about it. There is no specific technique for doing that. It entirely depends on which method you are using and who else is there in the tree along with them. Does that answer your question, Kushi?
Okay, so means basically we guess, we put a guess.
It's
We
always
can
a
say.
guess. Any tree making is always a guess. There is nothing else that you can do about it.
Oh, yeah, yeah. Okay, sir. Thank you.
Okay. Okay. Chinmay, do you have anything? Okay. Chinmay says he doesn't have any queries till week 3. Okay. Darshana, can you now put your question?
Good afternoon, sir.
Good afternoon, Darshana.
I wanted to ask about the query from the week 3, revising Mendel and introducing population genetics.
Hmm.
So, in that, you mentioned about 3 laws of Mendel.
Hmm.
So, the first one was the law of dominance.
Hmm.
Actually, I read in some books that it is not considered a law, it is actually principle. So, what is every law is having some pros and cons and some exceptions. So, in that way, how can we regard the dominance as principle and the other two laws as the real laws?
I mean, look, I don't really distinguish between these two very, rigorously. Okay.
Okay.
Typically, principle, one can say that if it is an observation coming from a direct observation, whereas a law typically, if it is coming from certain assumptions and from those assumptions you have ended up doing something and therefore you get a statement. So, in the context of Hardy-Weinberg, think about it.
Yes.
You have first made all kinds of assumptions about Hardy-Weinberg, right?
Yes.
And then from there you are getting a consequence. The consequence is that the allele frequencies will not change and there will be a certain relationship between allele frequency and genotypic frequency, correct?
Okay. Okay.
So, you are not getting it straight away.
Yes.
Whereas, in the context of the Mendelian thing, there basically Mendel sees it for a long time and then comes to the conclusion that, hey, if there are two alleles, then one is going to be dominant over the other, and this is how do I define which one is the dominant? The one, if I do a pure cross, then the one which will be there in F1, that is the one that I am going to call as dominant.
Yes, sir.
Okay.
Yes,
So,
sir.
there you can see that it is more taking some observations and making sense out of it.
Yes, sir.
Whereas Hardy-Weinberg is more starting with some assumptions and then seeing what the consequences of that assumptions are. So,
Yes,
this
sir.
is one way in which you can kind of distinguish between the two, but honestly people do not make this finer distinction all the time. So people do call them 3 laws of Mendel.
Yes, sir. Okay, sir.
So, yeah, so it's not if you are doing a philosophy of biology class, then it will be very interesting, but generally people use those interchangeably.
Okay, sir. Okay, because sometimes while in subjective questions also, this thing is being like, they are not.
Really?
Yes, yes.
They are asking the distinguish between law and principle.
Yes.
My
So,
goodness.
so they are like they are calling us and they're asking, you don't know that these three are not laws. This is the principle. You go and see the
Well,
book.
I can, there are books I have read in which Hardy-Weinberg thing has been called a principle. And then there are other people who have written pages after pages to show that it is one of the most important laws.
Sir, no, sir. Actually, I was mentioning here about the law of dominance. So it is not regarding a
No,
law. It
no,
is a
that is,
principle.
no, no, I agree. But what
Yeah.
I just now told you, by
Yes, sir.
that token, Hardy-Weinberg should be a law, right?
Yes, sir. Yes, sir.
But what I'm
Exactly.
saying is that I have read books where they have just called it Hardy-Weinberg principle.
Yes, sir. Yes, sir.
So this distinction is not very, rigorously maintained in biology for sure.
Yes, sir.
ठीक है?
Thank you, sir.
Okay, great. Yeah. Binish, are you there?
Hi, sir, I'm here.
Hi, Binish.
I'm
Do
at
you?
work actually.
Oh, right. You
So
are in the Middle East, right?
yeah, Middle
Uh,
East. So I'm working today. Yes, and it's only 1:41 p.m. here in Saudi Arabia.
okay.
So yeah, I'm going through the course. It's hard for me because my memory doesn't help me. I think I'm too old now. So very interesting. I am also checking a lot of things which you referred, but equations, everything doesn't work for me. It's very hard for me.
Yeah, you can neglect
One
those also. You'll still get a lot even without going into the
Yes, a
equation.
lot of information. And one thing I just want to say, which will help also others.
Hmm.
I have worked with the butterfly heliconius. I have worked with them closely.
Mhm.
So we call it as postman. Postman is the name we call it. So when I was working in a butterfly garden in Sharjah, UAE, I was in charge of it. So we get pupas from South Costa Rica, specifically from Costa Rica. All the pupa look same, but when they emerge, it's all looks different. It's a very slow flying butterfly, and I see when you explained it, the markings are different, and how they live and everything else. So they're very delicate butterflies. So it's really helped me on your topic. And I saw a paper related to that also earlier about these markings and everything, which you were mentioning. It was very interesting.
Thank you.
That's all.
Thank you.
No
Okay.
questions because it takes time for me to learn and understand a lot.
Sure. I mean, you
Thank
can
you.
also put it on the forum. Anish is regularly checking the forum. So whatever question
Okay.
is coming there, he is answering them in the forum as well.
Okay. Thank you.
Yeah.
Thank you.
Thank you, Binish. Samiksha, do you have anything? Samiksha, are you there? Okay, so Samiksha says that she doesn't have anything. Deepak, are you around?
Yeah, I'm there. I have no questions to ask right now, but I'll
Okay,
keep following up with you, and I like the way you explain. So
thank
we
you.
will, yeah, I'll stay in contact with. I'm thankful that I'm getting regular updates from email, but lately off late I'm not able to follow up with it. But I'll keep in touch with you.
Sure, thank you. There is one question from Akib saying about altruism. Does evolutionary biology provide any answers on this? So I just last week recorded my lecture on altruism. It's a slightly longish lecture, 1 hour lecture, where I talk about the various theories that exist about altruism and what works and what doesn't work, and examples, counter examples, pretty much a little bit more detail than what I probably intended to, but I have done a reasonably thorough job of it. So you have to wait till week 7, Akib, to get that answer. Okay. Okay. Darshana has written Epigenetics and its Mysteries. Yeah. Darshana, you have anything special to say about the mysteries of epigenetics?
The example that somebody was explaining about the monarch butterfly or some experiments of the pupa, they told that everything was looking same, but when they emerged, it was like it was a differential kind of phenotyping.
Oh, in
So
that context.
yeah, in that.
Okay. Great. Okay. So we are done with the direct questions. So if you guys have any other queries, comments, anything, feel free to ask, feel free to type in the chat box. Yeah, Abhijit has joined just now. Abhijit, you have anything, any specific query in your mind? Abhijit, we couldn't hear anything. Perhaps there is a microphone problem at your end. Okay. Abhijit says nothing from my side. Great. Samiksha says, can I ask about career related questions? Let's wait a little bit. Let's see if anybody else has something directly related to the course, because after all, that's what I'm supposed to answer, right? And if nothing comes related to evolution or course, then we can go for it. Okay. Darshana says she likes the explanation of population genetics. Thank you, Darshana. Thanks a lot. So Akib says tree of life. I read that it's branched at the base due to horizontal gene transfer. Do you want to add anything? I mean, branching comes from vertical transfer. Branching doesn't come from horizontal transfer. What horizontal transfer does is it ends up connecting them in a what is known as a reticulate pattern as opposed to a pure branching pattern. In other words, if you have branching, then any given point offspring node, child node, is going to have only one ancestral node. Whereas if you end up having horizontal gene transfer, then one child node can have multiple ancestral nodes. That is the only difference. So, I mean, there is nothing particularly nice for me to add over there. I mean, like crossing at the base, then branching. What do you mean by crossing at the base, Akib? I'm not 100% sure I understand. Okay. So Akib says he will ask in the next session. No problem, Akib. No problem. Think it through and we'll talk. Okay. So if nobody else has anything, Samiksha, just ask whatever you wanted to ask. Okay. Sadhana has just now joined. Sadhana, do you have any queries for me? Sadhana Mishra, are you there? Okay. So Samiksha has written, I have completed masters in microbiology. So what opportunities except PhD are there for me. Are you asking opportunities related to microbiology specialization or opportunities in general? Specialization would be better. Okay. So, one thing of course is related to teaching. Many schools and colleges, they would really like somebody with a microbiology background. Another thing might be in very entry level positions in some pharma or other kinds of industries which use microbiologists, but at master's level those will be very basic entry positions. And apart from that, microbiology specifically, I don't know, I can't think of anything very special. Industries, teaching, if you have a medical microbiology kind of a specialization, then some of the pathology labs might be interested in hiring you. You might have to do a short course on medical pathology things, but yeah, many hospitals and labs, they want microbiologists for doing the routine testings. Sometimes yeah, no, this is for the testing they might want to do, but that's that's not a very high paying job, unfortunately. I've heard that most hospitals pay rather smallish amounts, so that is that. And otherwise, if it is not related to your specialization, then of course there are so many other avenues that you can think of. So yeah, I'm sorry, I don't have much along that axis. Yeah. Sudarshana wanted to say something. Aradhya says, due to college reporting and shifting, I missed one of my assignments. Can I still give the exam? I don't know this. This is, this you have to ask to the NPTEL folks. What is their policy around it? See, my job is to just set the question papers and give it to NPTEL. All these administrative aspects, NPTEL takes care. So just write an email and they are very responsive. They will give you the response. Okay. So Akib asks, do you think humans could eventually, do you think humans could eventually speciate again, or has globalization slowed or stopped that possibility? I mean, look, globalization, industrialization, all those have kind of accelerated the time scales. But if you look at quote unquote evolutionary time scales, I don't see why evolution cannot lead to a form of human beings which will be so different from the current Homo sapiens that you and I represent that we will not call it a different species. So short answer, I think it can be done. Time scale might be a little longer. Uh, as a beginner in BS biotechnology, how should one start their lab journey and navigate this like an explorer who wants to seek and delve research prospects, out of box, out of syllabus, beginner tips, guidelines, your views? Oh my God. Look, I mean, if you are trying to get a PhD, then do a postdoc, and then do a formal journey the way scientists do in order to get a job, then you obviously have to go by the rules, play by the book, so to speak. On the other hand, if you want to be an explorer, if you want to have fun, then you can do whatever you want. The only thing is that in order to do whatever you want, you will also require some expertise. You will also require access to machines. You will require access to consumables, chemicals and all that. And all that stuff can only happen in a formal setting. Doing that in an informal setting is very, very difficult, until and unless, of course, you have crores and crores of money, in which case probably you won't be asking this question anyway. So my suggestion is that you get into a lab and see, different labs have different cultures. Some labs promote exploratory trips for their students. Other labs are very frowning upon it, and these are the protocols, just follow the protocols and don't use your head at all. So obviously, if you are interested in thinking on your own feet, thinking slightly out of the box and doing your own stuff a little bit, then try to get into a lab which is more conducive for doing this kind of exploratory work, and then essentially negotiate with the PI. See, I will tell you one thing, almost all labs, students get quite a bit of time after doing their daily work. And if you are a real explorer, and if you sit down on the bench and think of a slightly different experiment, and you do that, you talk to your PI, most PIs are probably going to be perfectly happy supporting you as long as you have done whatever your regular quota of work is. So I think, again, there is no secret sauce. That's the whole problem here. Okay, everybody figures out their own path, and everybody does it in their own way. I can't really give you any better tips or better guidelines on this topic, simply because none exist. My views on it, yeah, so definitely you should do it, no problems. So Abhijit, in response to Aradhya's question, is saying that NPTEL is best of 8, so you can. I don't know if this is correct, then Aradhya, that solves your problem, but I still think you should get a confirmation from the NPTEL folks. Achha. Kritika has a longish question. Says that under the topic of descent with modification, discussed about Carbonaria and Typica forms, correct? How do we define that their wings colored change, and it wasn't simply due to being higher visibility to predators, one form significantly reduced due to being preyed on and the other? Pritika, I don't think I understand your question. Can you please unmute your mic and tell me what you have in your mind? Pritika, are you there? Kritika?
Sir, she replied in the message box.
Yeah, what has she written?
She's at library.
Oh. Okay, but I don't understand what exactly she is saying. How do we define that their wings colored changed? Definition, this is not a definition, right? Are you trying to say how we infer? Kritika, can you just write a little bit explanation? Yeah. Okay, she says, I'll explain my question in the next meeting. Okay, perfect.
So she's saying that how do we know that there was a mutation and it wasn't just those passing on their genes, the white color or the black ones? She's asking that, I guess. Like it wasn't like, let's say there were 10 in one color and they reproduced exponentially, and that's how we got. How do we get the mutation part? I think that's what she means.
No, I don't think that is the question. I don't think that is the question. I mean, what the information we have is that the relative frequencies just change. There existed both forms before industrialization. Yes, we know that, correct. So I am asking, how can we say that their wings color changed over time? No. See, pre-industrialization, the frequency of the Carbonaria form was extremely low. And then when industrialization happened, very soon the frequency of the Carbonaria form increased quite a bit. I mean, it almost became total, close to 100%. And we say that it is because of predator-prey interaction. Okay, there is no question of saying it is not because of predator-prey interaction. The whole idea is that it is because of predator-prey interaction. Does that make it clear?
Sir, I think we studied in like species change. So she's saying she thinks that the black one, the former typica changed into the other in that way.
Nobody says that former typica changed into the other, no. I never said that. All I said is that the frequency changed from being in favor of typica, which basically means higher frequency of typica, changed to a higher frequency of Carbonaria. That is not saying that the white colored moths became black colored. Okay, the whole idea is that the white colored moths, they ended up facing higher rate of predation, and because they ended up facing higher rate of predation, therefore their numbers went down, their fitness went down, and the other thing went up. Correct.
Sir, but we studied it under speciation. That's I guess.
Sorry, who studied it under speciation? I didn't teach you under speciation here. I haven't gone to speciation yet.
I mean, it's a species change. We learned that under that species change.
Species change, matlab kya? What you guys learned is that the frequency changes over time. Isn't it? The frequency changes over time. That doesn't mean that the white becomes black or the black becomes white. Okay. That is the critical thing. Yes, exactly. That is the data, that there was a change in the frequency. There is no observation which says that the white became black. And then, after the pollution reduced and the frequency again changed in the other direction, there is no data which said that the black becomes white. Okay. Does that make sense?
Yes, sir, but we just learned it under like we asked the question, do species changed? And you said yes.
Do species change? That means that, see, the population property change, but that doesn't mean that one species became other species in this particular context. They still remained bison betularia, right? See, the whole question was, I mean, think about the context in which I said the whole thing. Initially, people said that, look, the properties of a species are immutable. A species cannot change. Species are the way they were made. And then we said that no, that is not true. A species can change while it is remaining the same species. Bison betularia was an example of that thing, right? But that doesn't mean that it changed to become something new. Not in that one. Okay. And how the change is happening? The change is happening due to natural selection, which is happening because of the background changing and the predator seeing and so on and so forth. No. This color change is not speciation, please. No, not at all. This color change is not speciation. If it were speciation, then it would have been given a new name, right? No, it's still bison betularia. And the white form and the black form can mate with each other and produce babies. Okay, so that's the point. Is it clear now? ठीक है, great. हाँ. Is there anything else from anyone?
Sir, there was a question above the feed.
Hmm.
The person was asking difference between homologous and homologous genes.
Huh. So homologous and homologous gene. Somebody is talking about Y chromosome shortening. Okay, I'll come to Y chromosome shortening later. Yeah, I am not 100% sure that I understand the difference between homologous and homologous. Typically, what happens is that when you have two genes which have a common ancestor, that's when you call that these are homologous genes. Okay. Whereas when you have genes which have evolved differently in different species and somehow they have come together in the same species, that is what is called a homologous gene. But I am not aware of too many examples of homologous genes. Okay, I have heard the concept. I do not think I am aware of examples. Okay. Take on Y chromosome shortening. I mean, what is the take? I mean, it happens, when there is no crossing over, but there is no recombination. So then essentially you get into a phenomenon known as Muller's ratchet, and due to Muller's ratchet, you can, you essentially end up having zero selection on that particular thing, and it shortens. So there is this assumption, or rather this prediction, that at some point the Y chromosome will become so small that it will become basically useless or non-existent. So that is that. In evolutionary context, can we combat its maleffects? You mean to say Y chromosome shortening's maleffects? Hmm. I mean, see, for anything, technically speaking, you should be able to combat its maleffects if you have the corresponding mutations in either the X chromosome or on the other homologous or the other autosomal chromosomes. Whether we will be able to do it or not, whether the precise mutation that we need will come about or not, that question I cannot answer. I mean, nobody can answer, right? So theoretically, is it possible? Yes. What is the probability? I have no clue. Nobody can calculate that probability. Will we end as a species in near future? Are you saying, are we going to go extinct or not? I mean, if the current world situation doesn't get sorted out within the next 1 year or so, I will say the probability is very high. We'll most probably have a massive, third world war and everything will just disappear. But otherwise, if you are asking whether we can evolve into a new species or not, yeah, sure. As I already said, there is nothing in evolutionary biology which is stopping us from evolving into a new species. So theoretically, perfectly doable. Practically, when will it happen? How will it happen? I don't know.
Sorry, sir, it was a mess right there. I was in bus coming back to college. That's why.
Yeah, you said that. No problems.
Yeah. Like, the confusion was that we thought that it's like the forma typica is changing into, even though it stays the same, that the species is same, biologically speaking. That's what we thought.
Okay, now I get the confusion.
The
Fine,
frequency, now we get it. The frequency is changing.
Yes.
Not
yes.
yes, sir.
So will this chromosome glitch, I assume that, yeah, you are talking about the Y chromosome thing. Will it affect evolution? Yeah, of course. Everything can potentially affect evolution. But what exactly the species will do about it, that we'll have to check. User, can you please tell us your name? It's a little irritating to call somebody user. Acha, Aditi. Oh, okay. You can change your name on Zoom. You know that, right? So if you click to on participants, you should be able to do that. Yeah, you click on participants, then right next to your name there will be 3 dots. Click on that, you can change your name. Okay. Hmm. Mutations are happening within Tibetan people. Yeah, I mean, why single out the Tibetans? Mutations are happening in everybody.
Clara, I'm just telling that it's it can end up as a new species because a lot of things are happening there. Like they are not traveling anywhere; they are staying there in one place for long. So
if that were
and
to be
the
the case, all the
and the
elevation then is a
oxygen.
different thing that they have some Denisovan ancestry and all that. But if what
Yes.
you are saying is correct, then far more than the Tibetan people, all these people living in these Polynesian islands, etc., they
Yes.
are isolated and have been isolated for a very long time. I mean,
Yes.
even
Yes.
some of the tribals in our own Andaman and Nicobar Islands, the Jarawas and the Sentinelese and all those guys, they have been isolated from whatever mainland India for the longest time. They have not speciated at all. So we don't know how
Yes.
long.
Yes. No, but the environment conditions is almost similar to the mainland people there. But in Tibet, there was a recent paper came where there's a lot of changes were happening with them.
Recent time changes.
Yeah. So recently came up actually. I'll find it and send to you.
Yeah, please. See,
Sure.
I know that in human beings there are changes. So for example, I'm going to talk about it when we do human evolution. There are these Bajau people. Uh, where are they? I think
Yeah, they
somewhere
are Polynesia. They are in the Polynesia.
the
Bajau. They can
Polynesians.
go underwater. Yeah.
Yes, for a very long time. And they have developed changes in the spleen which allows them to do that. But even in those cases where you have proper examples of changes, even their speciation, we are far from it. With the Tibetans, the point is, see, recently, well, not recently, for a very long time, the Chinese have been doing lot of developmental projects in Tibet, right? So because of which the Tibetans are not as isolated as we think they are. I mean, even if they are not going out, the Chinese people, mainland China people are coming in. So I don't know how much isolation is there in Tibet. We have to check it.
Yeah, yes, sir, you're right. I had a talk with the Tibetan people recently, and they are not allowed to travel anywhere else. So they have a small community like
They are not allowed to go, but others are coming in.
coming in. Yeah, Chinese are running some industry and some kind of business happening there.
Correct.
That's true. But
Correct. So as
among
long as there is mixing happening,
yes,
it will work, no?
yes, you're right, sir.
Okay. Anything else anybody would like to comment? Vijayan, I think you came in a little late. Do you have any queries? Vijayan, are you there? Hmm. So Abhijit is saying we generally gain features which are useful and lose which are useless. So in that context, did we lose vitamin C gene considering it's preserved in lot of monkey species but lost in apes? See, the point is that many a time we don't really gain features simply because they are useful. So for example, the ability to photosynthesize would have been absolutely awesome in humans. The entire world's food crisis would have disappeared, but we haven't got that thing, right? Similarly, many a times we end up getting some things as part of our evolutionary heritage. We didn't really lose it because it's useless. We lost it because we never had it to begin with. So in the context of vitamin C synthesis in particular, it is thought that the ancestral form ended up losing it at some point due to whatever reason. Doesn't necessarily have to be selection, might even can be genetic drift due to a founder effect or a bottleneck effect or whatever. But once that happened, after that, for whatever genetic reasons, we have not been able to go back. So that is that. Okay, just because something is lost doesn't mean that it was useless. Just because something is gained doesn't mean that it is ultra useful. It's a little bit more complicated than that. Okay. Does that answer your comment? Okay. Okay. If there are no further comments, can we call it a day? Can we finish? Or you guys have something that you would like to quickly put in? Thank you, Aditi. Yeah, it was nice talking to all of you too. Thanks,
Thank you,
Manish.
sir.
Thank you, Anatya. Chalo, so Karthik, we are ending this session. So shall I end the meeting? Karthik, are you there?
Yes, sir. If
Huh.
there is no question, means we will
Yeah.
wind up. Sir.
Yeah, let's wind up. Thank you, Karthik. Thanks a lot.
Thank you, sir. Thank you, sir.
See you people. See you everybody in the next one. Okay.
Yes, sir.
Bye.
Thank you.